Okay, good. Then I will suggest we will start now because there are a few words for the actual workshop that we want to say you. First of all, thank you, dear participants, for being with us all this week by our workshops and all the theoretical parts that we discussed before. It was a lot of fun, and we are appreciating a lot your activity and all your questions and suggestions in the feedbacks that you gave us. So thank you very much once more. Today we're going to start with the workshop day 2 session. And what we are going to analyze today is something else as we analyzed yesterday. Maybe some of you saw already the data sets that we provided you with, but I would like to say a few words about the data sets that we're going to analyze today. So basically today we are analyzing the other side of single cell. We will dive into the immunology, and we will have to deal with some immunological cells. And we will not only analyze the RNA signal on itself today, but we will also analyze the protein signal of these cells. So to remind you a little bit what we are talking about today, I would like to share you the screen from the presentation from day 2. So if you are participating on that day, you may recognize these pictures. Let's imagine that we are having some sample, for example, blood or some other cell suspension, and we are just working through our regular workflow. And in the next part that we are doing with this sample, we will label the sample with special antibodies that are directed against some cell surface proteins of the cells in this suspension. So these antibodies are so special because they don't have some fluorochromes or other enzyme molecules that can provide you with the color on staining. Now they have something else. They have an oligonucleotide sequence that is connected with this antibody. So as you can see, this sequence has also a poly-A tail. So if you remember, messenger RNA, for example, had also a poly-A tail that could be targeted. And the same is here. You have a poly-A tail, that barcode for each antibody, and the PCR handle. So this oligonucleotide will allow you to target this antibody if it's binded to the protein, and then to do the reverse transcription PCR out of this oligonucleotide that you are using. And at the end, you can combine it with the normal messenger RNA targeting techniques like single-cell RNA sequencing, and you're getting 2 libraries. You're getting one gene expression library out of the messenger RNA molecules that you have in your sample, and the second library will refer to the antibodies that binded the cell surface. So that means we have the second library with cell surface proteins. And that's very interesting, of course, because you are getting 2 different inputs, one out of the messenger RNA, and the second one we are using is the same from the cell surface proteins library. So today we are going to analyze a sample, a data set out of the cord blood mononuclear cells. So it's something very special, but it is nothing less as blood that was taken from cord. And then all the mononuclear cells, that means especially all the white cells that are in this blood, they were isolated and then prepared via CytSec workflow as you see it here. So what I have done with this data set that we will analyze today, I trimmed it a little bit. I have left there only few cell populations, so the analysis is not too complicated for us today. And I downsized the sample size out to the 2,900 cells. So that is something that we were going to do today. So basically, let's dive into the practical part. So first of all, it depends, of course, what you're working with. There are 3 different possible scenarios. The first one, you're working in your RStudio. So if you're working in your own RStudio, you just need to download the code as it was written in the introduction on the GitHub. So this is the code to download the files today. And you will see that in this archive that we are providing you, there are 3 files. 2 of them are CSV files, and the third one is a Markdown file. So the second scenario, if you are working in the Docker you can open the Docker from the yesterday and then just type into the console the code that I sent into the chat. So basically, you will download the files for today's workshop into your Docker. And the third scenario is if you are working in the R Studio on cloud, you just need to open the project and you will see the files in it. So I think there is only one person that is going to work today on the R Studio Cloud. So I know that this part is the most critical one from yesterday. So that's why we are going to do it like this. You will raise your hand up if you are ready with all of the files. If you see 3 files in your files Docker here, AB accounts, geeks, and also the markdown file. So please raise your hand when you are ready and if you are ready, because I just want to start with when all people are inside of the session, okay? And please don't unraise your hand so I can see who is with us and who is not with us. So because we had a lot of questions yesterday with, yeah, how can we proceed, and the one function doesn't work or the other one doesn't work. So let's start all together today so I know that you are with us. So basically, you need to open this RMD file, SC course workshop day 2 RMD, and you need to have it in the window so you see all the comments that we walk through today. And that would be the starting point. So we have all 3 files, AB counts, geeks counts, and also single cell course workshop data. So I see there are plenty of people that didn't raise their hand. Are you having some problems with downloading or? Okay, I see. I see also the video. Good, good. Is anybody there that doesn't see these 3 files or haven't downloaded them? Write me in the chat in Zoom so I can see you and then we can resolve your problem. So I see that nobody wrote, so I assume that we can. So if you can see the file, it may be helpful if you refresh the folder here. So you can go back and then you can go once more into your folder and you will see the files here. Okay. Yeah. I can see you in video here, so it's not a problem that you can raise your hand. Thank you. So as I said, sometimes it just doesn't refresh here. So sometimes you just don't see these files here. You just need to go back and then once more, and you will see the files there if you downloaded it properly. I send the code once more. For all the people that are watching us in YouTube, we have uploaded also all the files into the GitHub. So you can just open your RStudio or your Docker and then just write in the code with the download of the files, and then you can just do it simultaneously with us. All the markdown files are also provided on the GitHub. Okay, good. So let's start. Basically, the first thing that we will do today, just say me if you see my display. Okay, I see you. Okay, good. We will just load in our packages. That would be the same packages as the day before. We will use CRAD. We will use also some ggplot packages. And this command here, the next one in the markdown, is also a helpful one if you are working, for example, in some other environments as UPita or etc. It helps to set the size to the plots. So with this command, you can set the size to the plots. And in this case, it will be 11 to 11, and it helps you just to scale your plots. So let's run the next command. We will lower the counts matrices. As you can see in this archive that I provided you, there are 2 files, the AB counts. This is the count file for the antibodies, and the geeks counts. So these files are nothing less. It's just a really big table where the information for each gene and each cell is contained within the matrix. So let's just run it. It might take some time till you have your count matrices loaded into the file. So we can just wait a little bit, because out of these matrices, you will have to make a Cred object. As Alexander explained to you yesterday, the package that we are working is a Cred package, and it helps you to analyze the single cell data in order that you will create an object with different assays in it, and we'll take a look at the structure furthermore. So let's create our Cred object, and the first one, what we will create is the one that contains the counts from the RNA file. So we will create the object out of the RNA information and store it into the variable CBMC. We are working with cord blood mononuclear cells today. That's why the CBMC is the variable that we are going to use today. Okay, so let's run the code. As you can see here, the command CBMC will give you an output about the file, and you can see that our Cred object is created correctly. We have 20,000 of genes within 2,390 samples. And everything is stored within one assay with the name RNA. So we have successfully created the Cred object with the RNA file. So please raise your hand if you created successfully your object, your Cred object with this file, whether it worked for you. So I see, very good, very good. I also see the raised hands in the video. Cool. So does anybody experience some problem with this in this case? Does anybody have some problems creating the Cred object? Okay, I assume not, as I see in the chats. And okay, somebody is reinstalling the Cred package. Yeah, it might take some time, but the whole markdown file is there, so you can just run through and follow in, okay, with the notes. Thank you. So as you can see, the function assays gives you the output of the present assays in the Cred object, and we only have one assay because we created the information out of the RNA data, and this assay is called RNA. So somebody wanted to unmute herself? Okay, not. You can even look into the structure of your Cred object. For example, here, when you just click on the CBMC as your Cred object, you can take a look on the structure, and you see that the CBMC is nothing less, it's just a big structure out of assays, and where you can see your RNA assay. There is also a metadata in this file. So metadata will contain some additional information about your sample. And there are a lot of other things that you can also watch through, but the most important two things that are stored within the Cred object are the assays that we are working with, and also the metadata file. So the next thing that we are going to do after we created our assay object, our Cred object, we need to load also our antibody data in it. And we don't want to load it into the RNA assay. So that's why we need to create another assay to store this information out of the antibodies. So let's create a new assay to store this information with the help of the create assay object function of Cred. And then we can add this assay to the previously created Cred object with the help of this command. So let's run it. Now, as you did just the variable, you created a variable that contains this assay from the antibodies that you loaded in. And then you're just assigning, with the help of this command, a new assay to your Cred object. So the question, as I see in the Zoom, how did the metadata file got into the CRED object? In the way when you are creating the CRED object, the function on itself creates a structure with automatically metadata in it. And we will take a look at the metadata so I understand which information is stored in that. But it's an automatical process that is created with the help of the create CRED object function. Okay, so it's just an algorithm of the CRED on itself. And the next question is, here, why we don't want to have the antibodies file in the CRED object? We want to have it, but we want to have it in the different assay. Because as I said, you have different assay, you have RNA assay and also the antibody assay. So for example, here, when you add the assay with the help of this command with double brackets, here you can assign a name to your assay. You can see that the output of the function assays of our CRED object is providing us two different assays now. We have an RNA assay and also the newly created antibody assay. Okay, is it clear now? Cool, cool, thank you. So that means now with the help of this command, we created the second assay that is named AB. So somehow my RStudio Cloud reconnected. Excuse me. It takes just a few seconds, so I can just go back inside. Maybe somebody walked into the project, and that's why it just dropped me out. So now I'm inside. Good. So as you can see, now you have two assays that are called RNA and also AB. And if you want to see the names of the antibodies that you have in your AB assay, you can just use the function row names with the help of your CRED object and assay AB. Yeah, because with this command with double brackets and double quotes AB, you can access your assay that is called antibodies or AB. So basically here we see nothing less than just the proteins that we were using in our antibody assay. And this would be also the protein names that we can use also for plotting, okay? So now we can visualize it with the help of the row names command out of the antibody assay. So the next thing that we are going to do is the usual one. We will do the quality control. As Alexander explained to you yesterday, the quality control step is essential at the beginning. You need to filter out your dead cells, and you need to filter out the cells that have too less or too much gene counts. So let's run the function here. This function allows us to create the percentage of the features that I explained through the mitochondrial features. Okay. Mitochondrial features are the genes in the mitochondria. And the point is when the cell is dying, it is just releasing a lot of the inputs that is stored in the mitochondria. So logically, the cell that is dying would have a lot of the mitochondrial transcripts in the cytoplasm. That's why we need to filter out those cells that have too much mitochondrial transcripts. So, and the next point is also has been done yesterday. You need to plot this information with a Vialing plot. So let's run this function. And you can see you have three Vialing plots. The first one shows you the number of genes of messenger RNA coded genes that are expressed by your cell. And you can see the Vialing has its mean value a little bit less than 1,000. And you see that you are not going much more than 2,500 genes in this case. The N-count RNA gives you the similar information, but in that case about the number of UMIs that are detected in each of the cells. And here you can see the percentage of the mitochondrial genes that are in your cells. And you see that the percentage of your mitochondrial genes is in mean value around 0.03. So that means 3%. And it's a very good value. Because for me, the personal cutoff would be around 10 or 15%. when I'm working with immune cells. So in that case, we are really in very good green area. Let's go to the next chunk. With the help of the feature scatter plot, you can plot two different features. So one on the y-axis is the percent mito. On the other axis is the N-count RNA. For some of you that have been working with FACS data, this plot is really very familiar because you are just plotting two features on itself, and each of the dots is the cell in our sample. So you can see that you plot the percentage of mitochondrial genes against the number of UMIs in each cells, and you see that the majority of cells is being clustering here, so it's a good sign. You just need to see the majority of the cells that are clustering in one blob here. And if you would have a lot of the dying cells, you would see a great population here that is having a lot of the percentage mito and low UMIs. So that would be the cells that you need to filter out. But in that case, we see just one blob, that all the percent mito are lower than 10%, and that they are clustering together, so it looks really fine. So I see a question in Zoom. Does that change depending on the source cells in the data set? Of course, it changes if you're working with even if frozen cells or if you're working with fresh cells, you might see some shifts. And for example, if you are working with cells that are producing a lot of RNA, like plasma cells or some active B cells or myeloid cells, you would see a shift to the right here because you would detect more UMIs in it. But that's why I'm just saying you need to look on your plots, on your all cells. And if you see, for example, some cell population that are definitely over 10 or 15 and just making this narrow cut on the left, so that would be definitely the cells that you just need to filter out. So do it more flexible according to the data set that you have. Look at your plots, look at distribution here, and also at the distribution there. So the next one is also very helpful because in this case, we are plotting the UMIs against the genes. So you can see here a nice line that is saying you have a lot of UMIs and then you have a lot of genes, and that's why it's nearby linear correlation in that case, and that's fine. It would be not fine if you see a population that is here on the downside of the plot. So that would be also something that you need to filter out. Okay, so all the quality control steps that we need to do are done here. And in this case, we don't need to filter out some cells. So that's why we can just leave it and work with our further analysis. So before we start with a regular workflow that we used yesterday, I just want to ask you whether you have some questions or whether you are keeping up with us, whether you are now at the step with the quality control is done. Just raise your hand whether you are with us or ask your question if you have some questions. Cool. Okay. So good. People are telling that everything is also working by them. Thank you for your feedback. So let's continue working with our file. The main feature now is that you are having a file that contains two assays. You have a file with the RNA assay in it and also the antibody assay additionally to it. And you can see it here, for example, when you call your Selector object, you see the active assay, the assay you are working in. It's its RNA assay in this case. And you have also one other assay presented, antibody. So the thing is, as you know from the workshop yesterday, we need to normalize and to scale our data before we start to plot and work with the dimensional reduction. And you need to do it twice here because you have two assays, and that's why you need to normalize and scale each of the assays on itself. So let's start with the antibody assay. And you can see that you can change your default assay by the command default assay Selector object into the brackets and then antibody. So you can name your assay here that you wanna use as default assay. So that means with help of this command, we are switching into the antibody assay. And in the next state, we can normalize and scale the data of the antibody assay. So let's just run this command and you will see this output if it works right. So this is the case when you're normalizing your antibody assay. You see that normalization method is CLR. It's another method that is used in the RNA assay. So maybe the question is why? The thing is, it lies in the preparation of libraries, of the antibody library and the cDNA library. The thing is, when you are preparing the antibody library, you are doing less amplifications as if you would do it with the messenger RNA library. That's why you need to use another method for normalization this data. You are using the CLR method and not the logarithmic one that is used for the messenger RNA data. That's why we normalize it with the CLR method, and then we can scale the data afterwards we normalized it. As done yesterday, so you see we are doing just small recap also what Alexander did yesterday with the subset of the hepatic cells. Today we are doing the same, but with the monological ones. So now we normalized and scaled the RNA assay. The next thing that we are going to do is to switch back into our RNA assay. So we are doing with the help of the command default assay cbmc. And now our default assay is RNA. Now we can normalize our RNA assay. So note that in this case, the normalization method is the log normalize in comparison to the antibodies. So that is something that you need to keep in mind when you're working with two different assays. So when you normalize your data from the RNA assay, the next step would be to find variable features and to scale your assay. So let's find our variable features. So maybe somebody could explain me, why do I need to find the variable features within my assay? You can unmute yourself or write into the chat. Come on, we are doing an interactive session. Don't be shy. Just maybe somebody wants to explain why we need to normalize to find variable features in our data set. What will we do with these variable features? So everybody is shy. Nobody want to explain why do we need to find variable features in our data set? So I see the first comment in Zoom chat is that the differential expression. Not yet. The differential expression will come after you cluster the data. So, and then I see the suggestions afterwards, clustering and high dimensional data. We need to know which features are significantly informative of cell types, etc. Yeah, you are just right, guys. Thank you very much for writing into the chat. We need to find variable features in our data set to concentrate our attentions only on that genes that have high variance across our cells. Because with a high probability, only these genes will explain us the diversity of our cells and will help us to determine the cell type afterwards. Okay, so that's why we are finding variable features in order to decrease the number of genes only to those that can explain the diversity of our cells. We are doing with the find variable features function, and the selection method in this case would be the VST, and features will be 2000, so that means we will find 2000 genes. And the next thing that I'm doing is an optional one. You don't need to do this, but I really like to do it on my data sets. Sometimes the output of these variable features gives you also some genes that actually won't help you will not help you to determine the cell type. One of those genes could be, for example, this ribosomal gene markers. They will all start with the name RPL and RPS. So what I'm doing, sometimes I remove this RPL and RPS genes from the output of the find variables function. because I know that those genes will just will not explain me the diversity of the cells. That's the first one, and second, they can even disturb the result. So normally I'm removing the ribosomal genes before clustering these cells with the help of this function. So first that I'm doing, I'm searching for the pattern that is beginning with RPL and RPS. So that means this function will return me all the genes that start with these 3 letters. And afterwards, I will just remove all the genes that start with these 3 words out of the variable features. So it's a little bit complicated here. It's a function that just removes all the genes in our variable features that are in the list of this markers remove variable. So when you can run this function, you will see that at the beginning we had 2000 genes. Out of afterwards, when we remove these variable genes, we are getting to 1990. So we removed all the ribosomal genes from the variable genes, and then we can even plot them. So with the help of the plot function, variable features plot, and you can label these genes with the second one and combine them into one plot. So the output, as you can see here, highlights you only the variable genes within your data set, and it gives you a first insight into your data. For example, here you can see that a lot of the genes are referring to the cytotoxic CD8 cells, like GZMK, it's granzyme K, or GNLY, or KLRB1, that would be, or TRDC, that's T receptor, T cell receptor delta constant chain. That all are just the genes that are referring you to the CD8 cells and other T cells. Or for example, MS4A1 is a gene that is coding for the CD20. So maybe some of you worked with characterization of immune cells and know that CD20 could be a marker of late B cell and also can be preserved in some plasma cells. Or for example, you can see here, IgLC3, it's just the gene for the immunoglobulin light chain constant 3. So that means these genes would described as plasma cells. So we see a little bit diversity here. We see some genes that are referring to the T cells. We see some genes that are referring to the B cells, and even some of them can refer to the plasmablasts or some plasma cells. So this would be the first insight in our variable genes. Now we can scale the data and run the first dimensional reduction with the help of the PCI, as we did it yesterday with the help of Alexander. You can display the result of your PCA visually. So it's just the first 2 dimensions of your cells plotted against each other. If you want to see which dimensions that are and which genes describe each of the dimension, we can go to the output of the R console. And you can see here that the first PCA will contain genes like LTB or CD69 or TRBC2 or LDGB, HP. So those are genes really that are highly expressed by the T cells. So on the other hand, we can see some other genes in the negative compartment that can describe also some myeloid cells. In the second PCA, we see also genes on one side that are describing a lot of the T cells. On the other side, we see AGHM, so it's immunoglobulin gene, CD79A, or once more CD20, so it's a B cell genes and so on. So just explore your data on each of the steps that you are doing, that you are processing, because it helps you to navigate through the diversity of the genes and of the cell types that you can see possibly afterwards. Okay? So you can even see that a lot of the genes are HLA-DRB2, so it's an MHC protein type 2, and this MHC protein is expressed by a lot of myeloid cells or B cells. So you can, out of these genes, you can even predict which cell types you will see in your data set afterwards. So let's go further. What would be the next step after we did the PCA? The next step is to know where to stop, because you computed with the function run PCA 50 PCAs for your data set. You will not need all of the 50 dimensions of the PCA to cluster your cells. So you need to have some techniques to say, okay, where I'm stopping, starting with 1 and ending with 50. Yeah, where is the cutoff? One of these techniques that can help you to determine this number of the dimensional reductions would be the elbow plot technique. So you can visualize the standard deviation of each of the PC that was explained by your PCA, and you can see that you have a lot of the standard deviation at the beginning, and then when you are going further in your dimensions of the PCs, you are gaining no increase of the standard deviation. So that means when you're looking at the elbow plot, the cutoff point should be right after the elbow, somewhere here. Because when you're going furthermore in your elbow plot, it is not necessary, because these dimensions that you will have will not explain so much the diversity of your cells. On the other hand, you need to know which cell types you want to have in your data set, because if you need to separate some very rare subpopulations of the cells, it might be that you will take a little bit more dimensions as usually. If you are not expecting some very rare cell populations, you can just cut off at the elbow and then saying, okay, 10 dimensions would be the number that I can use for the further dimensional reduction. Okay? Even to specify it even more, you can visualize the genes of each of the dimension that is calculated by the PCA with the help of the heat maps. So this is the function that you're gonna run next step. You see here we've just generated a lot of plots for each of the dimensions of the PCA, starting with 1 and ending with 20. And you can see, for example, in the first PC, all the genes, you have really four areas of the expression, where you would have here the positive of your PCA and here the negative of the PCA. So as you can see, for example, here we have a clear cut for our PCA1 for the positive genes and also a clear cut for the negative ones. And that is the ideal picture of the PC, okay? That is something that is really very nice. And if you can see, for example, when you're going further in your PCs, this ideal picture just vanishes. You will see that, for example, in PC3, you are not longer discovering those 4 areas on the heat map. You can maybe discover here the negative ones, but the positive ones are not so clearly separated, and so on and so on. So we said with the elbow, our cutoff would be around 9, 10, 11, where the elbow ends, if you look once more into the plot here. So here would be the cutoff for our dimensions. So that's why we can take a look at our genes that are here and just try to think whether they make sense or not. Actually, they make, because, for example, here CCL5, it's a cytokine that is released by a lot of immune cells that can be described as T cells, and that's why we are gonna prefer to use also these genes in these dimensional reductions too. DASP2, for example, is one of the genes that is involved in the negative feedback regulation of the activity of the T cells, so it's also a nice gene to have. You see here in the complex CD8A, also some other genes. So for me, it looks fine. For me, I would use this dimension and just make a clear cut after this dimension, and I won't use any other, because you see, the furthermore we are going to 11 or to 12, you're just losing the ideal picture of your PCAs. In 12, you don't see even anything here, so can help you to discriminate between the cells. So that was our method to determine the numbers of the PCs to use. As Alexander said in the Zoom, so much nostalgia. Yeah, it really is, but it helps every time, really. You need to check all of your PCs every time you are working with data, because sometimes you take too less or sometimes you take too much, and it can affect the results that you are going to get afterwards. So as I said, we set the cutoff at 10 dimensions. As you can see here, we are running the next function, the neighboring function and clustering function. And the number of teams would be from 1 to 10, as we said before in the cutting off of our elbow plot technique and dim heat maps technique. So let's run these functions. And as you can see, the number of the communities that is computed now are 6, with the help of the resolution that is pointed at 0.5. This parameter is one that you can change every time, because this parameter will just increase the number of clusters or decrease it. It might be sometimes helpful to play a little bit with this resolution parameter to see what clustering works for your data set for the best. Because you need to know all techniques are very flexible. Yeah. Sometimes when you're working, you set the resolution to 0.5. Sometimes you see that 0.5 just works too robust, and that's why you are going to increase this resolution. So that's why answering the question in the chat, what does the resolution refer to? As I said, the resolution refers to the depth of the clustering in your cells. So with increasing the resolution, you will get much higher number of communities, and with decreasing it, you will have less communities. But it's not the case that all of your communities would be different cell types. So that's why you need to play a little bit with this parameter. Which clustering method is used in this function? As I know, it's a KNN neighboring finding and then based clustering on this KNN matrix. And as you can see here, the output gives us just 6 communities that will be in our data set afterwards. The next question is, what is the max number for resolution? I wouldn't prefer to go more than 2. Really, I'm always actually, when I'm analyzing T cells, I'm working with 0.5 till 1.2, not more. But sometimes it helps you to increase it a little bit more. Because as I said, if you put it too high, then we will have like 12 clusters that may be not separated by the cell type. Yeah, you will have like in one cell type maybe 5 clusters that doesn't differ much from each another. So that's why you need to every time to recapitulate which resolution you take for your data set. But for me, works 0.5, 0.7, 0.8, also very nice when I'm working with T cells. So the next point, after we calculated the numbers of the PCAs, after we calculated the clustering and the neighbors, we will do the dimensional reduction. The first one that we will try would be the t-SNE. So the t-SNE does nothing else as to dimensionally reduct. So that means to compress all your multi-dimensional file into just 2 dimensions. When you did it, you can run the next function and take a look how really shitty it looks, because you can see that I'm very biased. I don't like t-SNEs. So that's why this is the output of the t-SNE dimensional reduction. And the major criticizing point on t-SNE, why I don't like it, is actually, you see it here, the distance between the clusters in t-SNE, like the distance between 0 and 2 in t-SNE, doesn't say you too much as in comparison to other dimensional reduction techniques. So with the help of t-SNE, I cannot say for sure whether my cells 0 and 2 are much more related as 0 and 1. And that is the major criticizing point in t-SNE. So that's why it's something that I would not prefer to use. So let's just delete the output and go to the good technique. Let's use the UMAP. So it takes a little bit. After the UMAP is computed, you will get this output. Now look at this beautiful picture. Just look at this. So it's, in comparison to t-SNE, it's just beautiful, really. You see a lot of distance between your clusters. You can say that 0 and 3 are much more related as 0 and 1. So that's why, please, my good suggestion, goodwill suggestion, use the UMAP as a visualizing technique, okay? So on this point, I would make a little stop and ask you whether you are following or whether you have some questions. Just write me in chat or raise your hand, and I will see whether you are with us or not. Cool, cool. Thank you for your raising hands. Good, 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 good. So let's move further then. Thank you, thank you. So we can start with visualizing a bit. And as I said, we are working with the cord blood mononuclear cells. So that means we are hoping to see a lot of different immune cell types within, because we selected only the white cells from the whole blood, and on top of it, we are expecting to see only immunological cells in that case. And it helps a lot to, before we are doing the find markers function, just to take a look into some general markers, yeah, to visualize some general markers that can describe you some types of the cells. We are not doing the hardcore immunology today. Yeah, we are not doing a subtyping of diversity of different subtypes of CD8 positive T memory cells, like central memory, effector memory, and so on and so on. We are doing really light immunology today just to see what diversity you could have. So that's why let's use only some markers here. For example, CD3 for T cells, CD4 for CD4 positive T cells, CD8 for CD8. CD14 is one of the markers that is expressed by myeloid cells populations, and CD19 as B cell marker. Let's run the function, and as you can see, I really like to use loop functions. It's a nice way to spare you some time, because with the help of this function, you are just looping the same plot by the number of genes that you inserted in this string. So as you can see, genes RNA is a string, is a vector that is containing gene numbers, gene names for the genes we want to plot. So it's 1, 2, 3, 4, 5. And with the help of this function, you're just looping the feature plot functions 5 times in order to plot 5 different genes. And you are looping it through your genes RNA string. So that means by the first loop, you're gonna get the feature map for the CD3. By the second loop for CD4, and so on and so on. So it's very nice thing just to loop your functions, to iterate it in order to make your analysis faster. And let's look to the output. For example, we will start with CD3. And as I said, in this case, CD3E is a gene that describes T cells. And you can see that a majority of cells in cluster 0, 3, and 5 are expressing this gene. See a little signal in 2, but it's basically nothing. And you see a little bit more in 4, and we will talk about it later. When you're working with CD4, you can see, for example, here that you have signal in 0, and you can have a little bit signal in 4 and 1. So in that case, we will come to the problem of the messenger RNA with CD4 and CD8. CD8, on the other behalf, you can see that it's a cluster 3 that is expressing it by the major point. CD14 as myeloid cell cluster marker is expressed by 1 and 4. And CD19 is expressed basically just by 2. So it gives you just a brief insight into your cell clusters that you can have and just navigate you to how to separate the cells better. So we can do the same with antibodies. Let's switch into our antibody assay in order to plot it. And here I visualized once more the genes that are stored in this assay, just to remind us what are the names of the antibodies that we used. So in this case, you can see I used some of the antibodies, namely CD3, CD4, CD8, and CD19. I did the same plots, but using in this case only the antibody signal, not the RNA signal. And you can see here, for example, this is CD3. So this is the antibody signals that you can see. So in comparison to the RNA signal, you can see that you are gaining much, much more in that case. It comes even more when you're using the CD4. Because in that case, when you're looking into the antibody signal, it's really strong. It's really strong in 0, a little bit in 4, and in the other clusters, it's just intermediate level. When you compare it with the RNA signal, you can see that it's almost nothing in RNA, but a lot in the antibody signal. So if you remember the day 2 that we spoke about, is that there might be a situation where you have the protein, but you don't have the messenger RNA, and it is exactly that situation. As you can see, a lot of antibody signal, but in comparison, much, much less messenger RNA signal. So it helps you even better to describe your cells in the sample. When you look into the CD8, it's the same picture. You have very strong signal in 3. In comparison to RNA, in RNA, it's less signal. And the same for the CD19. You have very, very strong signal in cluster 2. So it helps you even more to understand which cluster is which cell type and to help you navigate. So, and as for example, our participant wrote in the chat, I can see the importance of using the antibodies too, as it shows you some are not showing much RNA, but the proteins are existent. Yeah, exactly. That's the thing that we are talking about. There might be situations you have a protein, you have a signal from protein, but you don't have a signal from the messenger RNA. Good. So we took a brief look into our data. We identified T cells that will be mainly located here at the bottom in cluster 0, 3, and 5. We identified B cells that are located in cluster 2. And we have some myeloid cells populations that are located here on the top of the cluster. So that's why we are going to prove our theory that we work now through with the help of the find markers function. Yeah, we'll unbiasedly find the markers in order to prove our theory that we work through now. But note that every time that you're working with 2 assays, you need to be sure in which assay you are working in. Because if you are working in the antibody assay and then you didn't switch to the antibody assay and try to find markers, it will find markers only in your antibodies. But the find markers function is needed to be used through the RNA assay. So let's just switch back into our RNA assay by the help of this command. And the next one, we will need to calculate the features for each of the clusters. So we are doing the differential expression analysis for each of the clusters against all the others. That is the thing that find all markers functions does. With only one modification. So you can see I'm using something very similar as I used in the variable features function. I'm looking through the markers that I can remove. And I'm looking specifically through the markers that are coding for the variable chains of the TCRs for the T cell receptors. So you can see TRAV or TRBV or TRGV or DV. So these are all the variable genes that are coding for the T cell receptor. Why am I doing this? The main cause is that you don't want to find your different TCR markers because they don't describe your cell identity. They just describe which TCR is expressed in each T cell. And that's why you just don't want to have these markers in the output. You don't need them because the T cell receptors genes don't help you to describe the cell on itself. And the same with the ribosomal genes. It don't help you to describe the cell type you are working with. So let's just remove these genes before we start the find markers function. Now we calculated the clusters and we did the differential expression analysis for each of them. And we can plot our UMAP in order to remind ourselves which cluster is where. So for example, 0, 5, 3 on the bottom, 4, 1 are here, and 2 is here. And let's take a brief look inside of the top 10 markers that are presented in the output of the find markers function. So as you can see, it's a table with a different marker in it. And for example, when we look in the cluster 0, you see interleukin-7 receptor, you see constant chain of the TCR, you see CD3E, CD3D, and CD3G. So it's just a very, very fine sign for you that you have T cells in cluster 0. And in that case, we looked that they are CD4 positive. So with high probability, cluster 0 is a CD4 positive T cell cluster. If we're looking further, the cluster 1 would be the one on the top. And you are having mainly genes that are associated here with myeloid cells, like LYZ or ThyroBP or LGLS1 and so on. So you see mainly genes that are associated with myeloid cells. It's proved our theory and so on. So it can be a nice homework for you. Just walk through these genes in the output and see whether you see some patterns, whether you can describe the cell populations by the help of the genes that you see in the output. If you cannot, it's not bad. You can help yourself at the beginning with the help of the web page that is called Enricher. And this is a nice web page where you can write in your genes that you has as an output of some function, like the ones that I'm writing now randomly into. So this, when you're writing the genes as an input here into the enricher, you can calculate afterwards the probability that you will have a certain cell type within your population. So you're just typing in your genes, you're going to the cell types. Sorry, just loading too slowly by me. So just a nice tool, as I said, to see which cell types you might have. And you can see here, human gene atlas, the probability that are the CD4 T cells is the highest one here. And it refers also to the genes that I wrote, because I wrote the genes that are called CD3, CD4, CCR7, and interleukin-7 receptors. So the genes would describe mainly the population that is CD4 positive T cell population. So nice tool really to help you, to navigate you. It will not work every time, because when you're getting too deep inside your data, you're subclustering a lot of immunological cells. It will not help you. But for general, like for example, to identify B cells or plasma cells, or even dendritic cells or plasmacytoid dendritic cells, it will help you. It's really a great tool to see which cell population you have. So let's move forward. We can visualize it, our genes that are variable across, that are differentially expressed across our clusters. And one of the methods would be to do a heat map. I like to average the expression before I'm doing a heat map. So I'm getting just one heat map on itself. And there is a function in CRAD that is doing you a new CRAD object out of the average expression. So with the help of this function, you're just creating a new CRAD object that will have the average expression of each gene in it. And then you can visualize this heat map with the help of the doHeatMap function using your new cluster average CBMC object. So when you're using this average CBMC object, you are getting only the heat map that regards to the average expression level in each of the cluster. So it might be a nice tool to look into your cluster markers. For example, here 0 is basically T cells, 1 is enriched only in the myeloid cell population, 4 as well, as you can see it here. Cluster 2 is basically B cell cluster, because you see CD20, you see also the immunoglobulin heavy chain for the M type, so IgM gene also in it. So it's really nice tool to see that you have just precise enrichment only in this case. And when you're moving just forward, just take a look into your markers. For example, CD3, you can see it here. It's CD8 positive, CD8A positive, B, and so on. So it's something that will basically tell you about the CD8 positive T cells. You can manipulate the size of your plot inside of the chunk, as you can see it here, with figure width in 12 and figure height 12 in that case. So you can just precisely predetermine how big your plot will be. You can change it, for example, on 8, and you will see how bad it looks now, or not so good. Let's say for 5. You will see what I mean in that case. You can see now you cannot even see the proper gene names. So just play with the size of your plot in that case and look inside of the output. So it's a heat map. It's an average. Firstly, it's an averaged heat map. So that means we just average the signal of the whole cluster. And the second, it means that you don't see the actual expression level. You see the relationship of the expression level of one cluster compared to another one. So you don't see any absolute values in the heat map, and that's a problem. It doesn't describe the actual expression level on itself. It's just describe how much one cluster is expressing it more as the other one. So how to avoid it, how to do our research clean. You might use some other techniques that can describe your genes and on the other point, described how much cells are expressed and on which level they are expressing it. And one of the methods that you can use, it may be a dot plot. Maybe it looks not so nice as heat map, but the dot plot gives you an output in form of dots. And you can see the average expression here of each of the dots. And you can see the percentage of the cells that are within the cluster that express this gene on a certain level. Okay, so that means that that in that case, dot plot might be a bit clearer and a bit better way to visualize your data in terms of good scientific practice, because you are showing firstly, the average expression, and secondly, the percentage of the cells that are expressing this gene. Because sometimes you might have only one cell that is overexpressing this gene, and then it pushes the whole heat map towards the higher expression. And in that case, with the size of the dot, we can see that it's not so important. For example, here you can see a very high expression, but a very small dot. So that means that this gene would probably not pass the good quality control. I see that somebody wrote in the chat, doesn't the relative expression data come from the average expression function? No, the average expression function just returns you the CRED object that has averaged all the expression of genes across one cluster. But this relative expression is coming from the doHeatMap function, because when heatmap is plotting a data, it just refers to the z-score. It's called the z-score. It's a relative scoring between the cluster on itself. So this relativeness comes from the heatmap on itself. So now we visualized all our markers. Now we know which cell types we have in our data set. We have but one small problem. If you look into the cluster 5, it's very uncertain what it is, because it expresses the T cell markers, but it's just clustering apart from all of the T cells. Yeah, it's here, it's this small population. So what could it be? In that case, you might test some markers. For example, here, we are testing the CCR7 marker, CCR7 and CD45RA. Those are the markers that can describe naive T cells. So, and when you are looking inside the Vialing plots of these markers, you see that cluster 5 isn't expressing it at all, but cluster 0 and 3 are expressing it on high level. So that means cluster 5 is not naive, but it's also a T cell. To prove the theory, we can take a look into the memory subtype markers. One of them would be the S100A4. And you can see that cluster 5 is expressing this marker much higher as 0 and 3. So as you can see here, it gives us an access to the possible identity of this cluster 5. Because as you can see in UMAP, it's clustering apart. Here we have all the T cells, but somehow this 5 cluster is a little bit different as 0 and 3. So that's why just look into your markers, look whether you have naive cells or whether you have memory subtype, and then you can even plot some of the negative feedback loop markers that are pointing towards very active T cells, and you can see that 5 is overexpressing it also in higher level. So cluster 5 would be something that is mainly memory subtype that is even a little bit exhausted. So, and I know that you may be exhausted too. So we are coming to the end, believe me. Okay, it's not so much that we need to discuss more. So, and now we want to rename our clusters 0, 1, 2, 3, 5, 4 into proper names in order to plot our data even better and in order to better working with these cells. So what would be the essential file where you can store your cell names? And now we are coming to the beginning of our course today. We are coming to the metadata file. So metadata file is a very, very cool place where you can store information about your cells, because it's built in this order that you have a lot of the row names. So each of the row is the cell, and you can see here that we have basically the barcodes of our cells as the names of the rows. So metadata is just a table where you have rows as cells, and columns would describe different information about this cell. And as you can see, for example, here, even our clustering information is stored here in these 2 columns that are called RNA SNN resolution 0.5, and also CRAD clusters. This basically 2 same columns that are describing the clustering. So we can store our clustering information also in the metadata. So let's add our metadata, our naming of the T cells into the metadata. I know that you did it yesterday with the help of the function rename idents. I'm just showing you another way how to store this information, how to rename your idents of the cells. And that would be the case if you, for example, use the metadata in order to do this. So how can we access the metadata? The metadata is accessed by the function cbmc add metadata. So you just need to write your CRAD object and then add metadata. And we are doing nothing less here as a loop. We are looping in a variable that is having a value from the first row till the last row of the metadata file. Okay, so that means we will loop through all the rows of our metadata file. And as you can see, I'm just pointing into in the next step into the if function. So I'm just saying if this row, and namely the column CRAD cluster, is equal 0, then write me in the other column that is named cluster names the value CD4 T cells. And then it proves once more whether this column, for example, if column CRAD clusters is equal 1. If it is the case, it will store in the column cluster names the name myeloid. If it's 2, it would be B cells. If it's 3, it would be CD8 T cells. So basically with this function, we are just going through each row in this CRAD clusters column that refer to each of the cell. And then we are just proving, okay, if you are having 2, we will write in another column the value B cells. If it's 3, we will write in another column that is called cluster names the value CD8 T cells, and so on and so on. So you're just iterating through your metadata. And now, when you run this code, you can check the metadata again. So let's scroll till the end, and you see a new column that is called cluster names, as we wanted it. And it stores a proper naming of the cells for each cell on itself. So why is it nice? Why is it nice to keep those information in the metadata? Because we can use this column to plot our cells. For example, here, with the help of dim plot function, I just want to do a UMAP. So, Gleb, do you wanna say something because you unmuted yourself? So Dima, can you please mute the participant here, because maybe he just did it on okay. Ah, okay, cool. So sorry. So basically what we are doing with the dim plot function, we can plot our cells with the help of UMAP. And in that case, note this attribute here that is called group by, and I can assign any column of my metadata file to group the cells. So in this case, I'm clustering according to the column that is called cluster names. So you can see now I'm plotting all the cells with new cluster names that can describe my cell identities. The other thing why it is so cool to keep this information in different columns is because with the help of the function idents, you can assign an idents to each of the column of your metadata file. So that means, for example, if I'm doing the function idents cbmc is equal to cluster names, I'm saying that the leveling of my cells, the assignment of my cells, will now take the information from this column. And that is the case where you can just switch between the identities within your metadata column. So it's a nice thing just to keep this information there, because for example, you can have a column that is called diagnosis group, and you can just easily do idents cbmc is equal diagnosis group, and then you can plot it according to the diagnosis group, or treatment, or etc. Okay, so that means this idents function assigns the levels of your cells according to the column of the metadata. Very useful thing if you are doing a lot of state analysis or a lot of diagnosis related analysis or treatment analysis and so on. So now we renamed our cells. Now it looks fine. We see that we have myeloid cells, B cells, CD4 cells, CD8 cells, and one of the clusters is called myeloid T doublets. So this is the case that you would have a lot of times, really, because myeloid cells are very happy sometimes to eat other cells. And that's why sometimes you would have either the doublet of these two cells inside one droplet, or you would have a lot of transcript for the other cells that is stored inside of the myeloid cell. And sometimes it is a problem to identify these cells. So just don't be very don't be very scared if you see some cell population that is combining markers of myeloid cells and the other cells population as well. Because it might be that it's just a myeloid cell, like monocyte or macrophage, that was eating a T cell, and that's why you see this transcript there. It's really very, very often. For example, here, if you look inside of the cluster 4, that is basically our doublet cluster, you can see that this cluster is expressing the information of CD14 expression. You see CD14 is positive in cluster 4, and CD14 is a myeloid cell marker. But on the other hand, it's also CD3 positive, and CD3 is T cell marker. So that's why sometimes you can have this doublet information or myeloid cell that is eating the other cell information. And don't be scared. It happens sometimes. It's okay. Just rename your cluster as a doublet or remove it for the further analysis, because when you have a cluster that's combining such two diverse cell types, it can affect your analysis in the future. So now when we look through the cell types, we can play a little bit with the antibody. We can use our antibodies even to visualize it better. So let's take a look inside of the different visualizing methods for the antibody signal. So for example, as we saw it before, we can do it with the feature plot with the help of the UMAP on top of the expression. So it would be a nice way to visualize our antibodies. For example, we can see CD8 expressed in CD8 T cells and also in the myeloid T doublets cluster. CD4 have the same story here. You see one cluster with CD4 and one cluster here in the doublet. The next point could be, for example, to visualize it with the help of Vialing plots. You can plot it here, for example, CD4, CD8, and CD45RA. This is a marker for naive T cells. As you can see, almost all of them are highly positive except of this T memory cluster. You see that the majority of this cluster is located in the downside of the Vialing plot. What is logical because we know that this cluster would be less naive and more memory subtype. You can do it with the rich plot. It's also a nice story. With the help of the rich plot, you can see a distribution of the antibody within each cell population. For example, if you look into the CD4, you can see that the major populations that express CD4 is CD4 T cells. T memory has also a big proportion here. And also myeloid T doublets would express some of the CD4 antibody in it. If you look into the CD8, you would see a distribution here in CD8 and also a little bit in T memory and in myeloid T doublets. So it's a way to visualize your antibodies that can show you the distribution of the antibody signal as it's usual, for example, if you're using some techniques like FlowJo and so on. So, and the last method that is very cool is, for example, to visualize this with the help of feature scatter. As I said, it may be familiar for some of you that were working with faxing, because with the help of this, you can really do it like in FlowJo. So you can plot one gene and another gene, like two features, like CD4 against CD3, and then see your major cell populations described by these two gene names. For example, here you can see a great population that is in yellow, and that is basically our CD4 T cells. And it's okay because it's CD4 positive, and also on the other subtype is CD3 positive too. On the other hand, the clusters that are negative for both markers will be here, and it's nothing less as myeloid or B cells, which is also logical. You can plot, for example, CD4 against CD45RA to gate only the cells that are more in naive T cell state. Or you can plot CD8 with CD45RA to gate only the CD8 naive cells, and so on. Why is it cool? Because if you work with the FACS data, you know that you can do gating with the help of the FlowJo. As I see the question in chat, what does the number on the top of the graph means? I have no idea what this number means. Sorry, I just don't know what it means. I never paid attention, even that there is some number on it.
I assume that it's like in regular regression analysis is the coefficient for line. So as much this as bigger is this number, it means that all of these cells on this medium diagonal line.
Basically correlation level or what?
Yeah. Mm-hmm.
Okay, good. I see that somebody wrote it also in chat. Yeah. Regarding the question of the myeloid T doublets, I will answer it after we look through the visualization methods, okay? So why did I say that this scatter plot is really a cool thing? Because you can work as if you were analyzing the FACS data. And the other one, you can even gate your cells as if you would do it in FlowJo. And you can do it also interactively here with the help of the function that is called cell selector. So basically, you can firstly assign a plot to the feature scatter of your two genes or two antibodies that you want to plot. And next, you just need to assign variable select cells to the function cell selector of this plot. So let's take a look what happens if you do it. The next thing that happens, you will have an interactive window that is opening on the right bottom that helps you to gate only the cells that you are interested in. And as you can see, in this case, we are analyzing CD4 against CD45RA. So CD45RA is a naive T cell marker. CD4 is CD4 T cell marker. So if we gate something that is very positive for both of them, like here, we can gate out the naive T cell population. So when we are done, we can just click done here. And now we gated out those cells. And you can see here we saved everything in the variable that is called select cells. So let's take a look, what does it contain? And if you run this code, you can see that this variable just contains the barcodes of the cells that you gated with the help of this method. So it's really, really very, very cool thing just to play around. And you see there are so much options. You are so flexible with these programming methods and with all the packages that you can use. Just dive into your analysis. And yeah, somebody wrote in chat, I feel like a child in a candy store with all these tools and options, lol. It's really nice. Yeah, I feel myself also when I'm working with single cell data. So regarding to the question about myeloid and T doublets, did you mention that they can be phagocytized in other immune cells? For example, they seem to have a lot of Cd4, Cd8. Have they engulfed some of these T cells? Yeah, that might be the case, especially if we are working with some tissues like spleen, where you have a lot of phagocytes that can eat the T cells. Or in this case, you have cord blood mononuclear cells, so it can happen that a myeloid cell just eats and phagocytes the other T cells. So, and the next question is, and this is something we can do even without the cell surface markers data, right? Yeah. You can just plot all the genes that you want. It doesn't matter if you're using the antibody assay or the RNA assay. It's just the method of visualizing, and we are familiar with this visualization method in matters of proteomics and faxing, and that's why I just wanted to show it here. But actually, you can visualize in this way any gene that you want. It doesn't depend what you are taking. Good. And the last but never least, we can talk about the new method that is coming with the new CRAD version 4. It was published. I don't know. No, it was presented in January this year. So as you remember, we clustered the cells with the help of the RNA data. But now in 2021, it is possible to cluster our cells based on RNA data and based on the antibody data. And that's very cool. So let's try it out. Basically, what you need to do is to switch here into your assay of the antibodies. And you need to run a dimensional reduction on the assay with antibodies. So with the help of this function, you can run the reductions, and you can store this reduction with the other name that is called antibody PSA. So you're running PCA on the antibody data, and that's basically the first dimensional reduction that you need to have. The second dimensional reduction that you need to have is, of course, the RNA dimensional reduction, what we did before. So the next step in terms of clustering would be to determine the dimensionality, the numbers of dimensions that we need to use for clustering. You are doing it with elbow plot. The first one would contain the information of PCA from the RNA data. So that is basically this that we saw before. That is where we set a cutoff for 10. And the second one would be having the information of your antibodies. And you can see that for antibodies, you have so less antibodies that you cannot build more than, in that case, more than 12 dimensional reduction. So this plot just stops by 12, and that's the highest number of the dimensions that you can calculate for this antibody assay. And in the next step, as you did it before for the RNA, now you are doing a combined weighted analysis. You are finding naive bores according to the PCAs of RNA data and antibody data. So you can select the reduction list that you are using. So we are using PCA for RNA and antibody PCA for antibodies. And here you can select the number of dimensions to use. I just used 10 for both. And then here you can store the results of this weighting with the help of this function. So let's just take a look. What is the result of the function? It calculates a bit. It may take a little bit time. The question, for example, in chat was, can you use different dimensions for each? For example, 10 for RNA and 12 for antibody. Yeah, you can use different number of dimensions for each of them. It just depends on you where you set the cutoff. Actually, I like to set it here where the elbow ends. So 10 for me, it was the case. But if you're having another data set, you just need to look into the elbow plots where to set the cutoff. So, and as we found the neighbors, but in that case, multimodal neighbors, we can now find new clusters according to these neighbors. Now let's take a look at the UMAP. So we are running the UMAP on this dimension, on this clustering results, and we can visualize them with the help of the DIN plots. So this is the new UMAP that is calculated according to the DNA, to the RNA information and according to the antibody information. So we have input of both of the types of the information. And as you can see, for example, here this cluster is clustering much more apart of the main cluster here. So you have a little bit more space between them in comparison that you saw in the RNA. And if you take a look in the previous names that we assigned before, so you can see that the CD8 cluster, for example, is clustering much more apart now from the CD4 and T cells as it was the case before. So now here I did nothing less just to plot the previous names that we used on this new DIN plot, just to look what is the distribution of cells now. So as you can see, the separation it's getting better, I think, because you separate the populations even much more far from each other. So you're getting some of the cells that are clustering now with CD8s, but are from the different color, so that may be even improvement. And afterwards, you can, of course, use other find markers functions and so on to see what are the markers for your new cluster based on the RNA and antibody information. So we can visualize for each cluster how important was the RNA information for it. So when you're plotting this RNA weight with the help of the Vialing plot, you can look inside to the percentage of the cells that were clustered with the help of the RNA information. So because basically this method that is clustering the cells based on both assays, it takes information from both of them and then compares how important this information is for each of the clusters. Some of the clusters might be better described with antibody data. That's why the RNA information would be just will be in background. On the other case, the antibody data is not so good, but the RNA information is better to use. So the algorithms choose how much should be the messenger RNA information weighted in comparison to the antibody data. And you can see it here. For example, the cluster 6 is the one that was basically explained mainly through the antibody information because the RNA weight is so low. And we can take a look what is cluster 6. So cluster 6 is here. And that is basically the one that will contain only the CD4 T cells, because it's here on the left side clustering with the CD4 T cells. So it's something that was chosen only on the behalf of the antibody information. On the other case, cluster 7 is one with the highest RNA weight. So that means the information from the messenger RNA was used mainly in comparison to the antibodies. And cluster 7 is this small cluster here that refers to maybe a small subpopulation of B cells that might be plasmablasts or on the way to the plasma cells. So a good homework for you, you can take this file and then just use the techniques for revealing the cell types that we used before, like plotting markers, finding differentially expressed genes, and so on, and find out whether this clustering is better as the clustering that you used according only to the RNA information. Play a little bit with it. It's just a nice homework to do. Use the methods that we already described to see whether this clustering is better as we calculated only on behalf of the RNA information before. So as I see there are some questions in chat, I would like to answer them. Instead of these two different type of data sets, can I do the same analysis for two different clusters in my RNA data set? What do you mean with two different clusters in RNA data set? Maybe you could specify this question, sorry.
So just instead of using the antibodies and the RNA, if I don't have the antibodies, then I use this analysis to see how some genes in a group of cells compare to the expression in the other group.
Yeah, so this is basically, if I understood it right, this is basically what you're doing with the find markers function. In the find markers function, you are trying to find the genes that are specifically enriched only in one group, or in some of the clusters that can describe these clusters better in comparison to the other ones. So this is what you're asking is probably the differential expression analysis, as I understood it, right?
Yes, this is probably
okay. So good. So we are at the end of the workshop day 2 today. So I hope that you gained some new skills with the help of this workshop. You gained some new insight into the immunological analysis and into the cell surface proteins analysis with the messenger RNA analysis. Feel free to ask questions if you have those, and we can discuss it together now when we are still in the meeting. And afterwards, the one thing that I would also like to ask you is just to fill out our evaluation form. We will provide you with links in the chat and also in the Slack. So this is basically the same as yesterday, but for day 2. It will help us a lot to improve our workshops in the future, and we are really reading all of the feedback that you are giving to us. So I see some questions here. What is the benefit of average expression? Upregulated and downregulated genes may sum to 0. Yeah, you are basically right. The average expression has only one benefit. It helps you to plot a lot of information very quickly. But the thing is, you need to prove every time the gene that you found based on the average expression, also with the help of the single cell plotting techniques, like, for example, Vialing Plot, yeah, or feature map, and so on. So the averaging of the expression helps you to take a quick look into your data, but keep in mind that sometimes you need to validate your markers and look every time into the particular genes using Vialing plots. That would be those here, if I may show it to you. So this one shows you really distribution for each and single cell in each cluster, and you need to validate all your markers with the help of the Vialing plots. Or, for example, you can use also feature map, because it shows you also on the U map where your cells are located, and you are using not the average values, but the absolute values of the expression in that case. So the next question is, can I look for surface markers expressed in my cells? Do you mean on the RNA or on the antibody level? Sorry, I don't hear you.
Yeah, I mean in the RNA.
In the RNA, yeah, of course. I mean, that is something that we did here, for example. If you specify the assay that you are working in, sorry, I come to this point now. Here, for example, I specified the assay as default assay for antibody. So that means every plot that I will be making will be in the antibody assay. If you want to do the same with the RNA, you just need to write default assay for RNA, and now you will plot everything that is in your RNA assay. And this is basically what I showed you before. So this is the RNA signal. And here in comparison is the antibody signal.
Yeah,
so
but I'm sorry, I think I wasn't clear at all. I was, my question goes more to how, is there a tool or a database where I can cross-reference my genes and then have the information of which of those genes are known to be expressed?
Yeah, I understand what do you mean. What could help you, for example, is this data set or like GO data set, where you can find all the proteins that refer to the group of cell surface proteins markers. So you can find this data set in the internet, like GO data set and so on, and you can download it from there, and then you will have like a large table containing the names only from those genes that are in the cell surface proteins category and in the cell surface proteins groups. And even more, for example, you can specify here in the find markers function the markers that you are using for your find markers function with the help of this features argument. So basically, if you want to find only the markers that are in the group of the cell surface proteins, you can load in the names of those genes that are in the group of the cell surface proteins and specifies them there. And then you will run only the analysis on those genes that are expressed on the cell surface.
Or like create like a data set like the antibody one.
Yeah, you don't need to create this data set. You have them as I said, there are databases that classifies proteins according to their expression where they are. And you just need to download from GO or from other website the data set that contains only the proteins, the names of the proteins that are expressed on the cell surface. And then you can load them in R and afterwards run the analysis only based on these genes that are on the cell surface.
Yeah, what I mean is that do I have to put that into the Selector?
Well, sorry, what did you say?
Do I have to put that information, that database in the Selector?
Yeah, you need to download it and then load it into the R, and then you can use it in the R for the find markers function. Okay.
Say R in the Selector.
Sorry, I didn't hear you. That's really very hard to hear.
I'm sorry, when you say R, you are talking about the Selector object that we are working on.
Yeah, probably in RStudio or any other environment that you're using. Yeah. Good. So the next question, as I see, is it possible to do single-cell RNA sequencing in fixed cells? Yeah, the next question is what are you using for the fixing your cells? Because it's very crucial for the RNA integrity. Maybe you can specify which solution are you using for the fixing the cells? In the chat, just answering. So because for example, if you're using like something formalin based and so on, like for tissue, it's not so possible to do a single cell RNA-seq out of the paraffin embedded FFP fixed cells. But if you're fixing, for example, in DMSO, like for liquid nitrogen freezing and so on, you can do it easily. It doesn't disturb the RNA quality at all. So the next question is, it's not my question, but is there anything you can suggest or seen used? I feel like the gene expression can be altered by the processing sometimes. What do you mean with the is there anything you can suggest or seen used? It's a little bit hard. With the fixing. Okay. So as I said, you can use, for example, for your PBMCs, fixing with DMSO, 10% in PBS, and then you can freeze those cells and you can tow them up after even 5 years, and you can do also the single cell RNA sequencing out of this. Acetone would be okay. I used already acetone for fixing some slides for the laser capture microdissection and RNA sequencing afterwards, and acetone didn't disrupt any integrity of the RNA. It was okay. But not PFA. Please avoid PFA because it will just destroy all of the RNA and DNA in your molecules. Good. So I think that we ran out of questions now, and it's very good point, I think, to cut it short now and to come to the end of our workshop.